brief intro. So I would give an introduction or basically follow up on the introduction that I started already on X-ray diffraction imaging. And that will be actually the only thing we do today because Tanuv and Anutama will then present their attosecond experiment tomorrow, together with Nishta and Vishal M, who will be presenting on X-ray coherent diffraction imaging. So my presentation today will be an introduction to this topic. So then let me start sharing my screen. Can you see my screen?
Yes, sir, it's visible.
Okay, perfect. Good. Then, this connects to the discussion of synchrotron beam lines and what comes at the very end of a synchrotron beam line, which is then the detector part, yeah, where the X-rays are shot onto a sample, and then somehow we have to detect whatever comes out of the sample, which can be electrons, ions, photons, yeah, some particles or some radiation that acts as the signal that we detect. And it turns out that these beam lines that are used for free electron lasers, for XFELs, are actually very similar to synchrotron beam lines. It's X-ray beams after all. The main difference being that the XFEL X-ray beams are much more intense and short pulses. So of course, this opens up new detection techniques we will get to in a minute. This is another recall. So in very general terms, what we can detect is photons that are scattered or emitted by fluorescence or by some other process from the sample, or we can detect electrons and ions. And in previous presentations by students, we have seen that by detecting electrons and ions, and actually detecting electrons ions in coincidence in using this cold rims or reaction microscope setup, is a very powerful technique where we could then extract the structure of molecules by Coulomb explosion imaging. But today we will focus on photon detection, and I think I showed this already. So one straightforward signal is to detect fluorescence light. So this is a straightforward experiment in the sense that the radiation from synchrotron or XFEL is sent on a sample, and then since this is highly energetic radiation, mainly inner shell electrons are excited or ionized. Then the inner shell holes are filled by Auger processes, or the excited states are de-excited by fluorescence emission, yeah, by just an electron falling into the hole and emitting the binding energy in form of photons, which produces these characteristic X-ray fluorescence spectra. So, of course, in order to resolve these spectra, we need some dispersive element. And since this is X-rays, we need a grating with a lattice constant on the order of angstrom, which is given by single crystals typically. And yeah, by using these bent crystals, then this is even more efficient in focusing the mostly isotropic fluorescent light, focusing like diffracting and focusing at the same time onto a detector to get the X-ray fluorescence spectrum. And this is For example, this can be used for very sensitive chemical analysis of a sample. This was a clay sample that contained a very small amount of cesium, and then in the fluorescence emission spectrum, besides many other elements that were present in the sample, then also a tiny fraction of cesium became visible as this characteristic line of the cesium ion. But so this is fluorescence. What now I want to get to is scattering or diffraction. This is mostly synonymous of X-ray radiation, and like the classical X-ray diffraction, which is also called XRD. X-ray diffraction is done from single crystals. Then we can use the nice periodicity of the crystal lattice and the Bragg diffraction condition to measure sharp diffraction orders and sharp peak patterns of the diffracted light. But most importantly nowadays is X-ray diffraction of macromolecules, like, of course, macromolecules with biological relevance, like proteins, which have both some crystalline structure, but also are highly disordered. So this is then presents itself as much more complex diffraction patterns. So just to recall the Bragg condition, I think you all know the condition for constructive interference of partial beams that are diffracted of these lattice planes. So in this textbook derivation of the Bragg condition, we only consider planes where the partial beams are refracted from, and then we can calculate what the path difference is for these different partial beams actually reflected from the planes, and this by requesting for having constructive interference at a specific angle of the light and requesting that the path difference should be multiples of half the wavelength, so that the multiples of the full wavelength to get constructive interference. Then we arrive at the Bragg condition. Now, if we take into account that crystal lattices are not just infinite planes, but they actually consist of individual lattice sites from which the light is diffracted, then this can be formulated in terms of this Laue condition for constructive interference. And the Laue condition, those of you who hopefully remember from your solid state physics course, what it says is that the difference vector between the incident wave vector, or the wave vector of the incident beam, and the scattered beam, the difference vector is this Q vector, that has to be equal to some vector in the reciprocal lattice, in reciprocal space. And we can also see that this is actually completely equivalent to the Bragg condition, essentially by looking at the geometry, like that Q relates to K, and where K is the absolute value of K in, which is equal to the absolute value of K out, because we consider only elastic scattering, so that the length of K in is equal to the length of K out. And then the angle between the two is 2 theta. So Q is 2K sine theta. And if that is supposed to be equal to a lattice vector, lattice vector in the reciprocal lattice, we have to remember how the reciprocal lattice is defined. Yeah, this is defined like the largest possible, the maximum lattice vector is, or no, any, so the largest lattice vector is 2 pi over d, and then any lattice vector is integer multiples of that, where d is the distance, the spacing between the lattice planes. And yeah, knowing that k in absolute value equals absolute value k out, and k remembering that the wave number, which is the absolute value of the wave vector, is 2 pi over lambda, then we arrive at exactly the same expression. So the point is just for you to understand this condition, the Laue condition for constructive interference of scattered beams, Q equals G, that is it's equivalent to the well-known Bragg condition. And why do I insist on this reciprocal lattice concept? Because there we can draw what is called the Ewald sphere in the lattice reciprocal lattice. So these points indicate lattice sites in the reciprocal space. And then we can draw our incoming wave vector k in and the scattered wave vector k out. And we can like shift around these the k in such that the error points exactly onto one site in the reciprocal lattice. Then yeah, if we now tilt around the k out to all possible angles, then only when k out points exactly on another site in reciprocal space, such that q, the difference vector here, is equal to some lattice vector, only then the Laue condition is fulfilled. And for this specific angle here between these two, between k in and k out, now the Laue condition is fulfilled, and we get constructive interference, and we expect to see a bright spot of reflected or of scattered X-rays. And yeah, so the experimental arrangement would be like this. I mean, k in, the wave vector, that's the vector pointing along the incident beam onto the sample. So if we draw the detector plane like this, and we should probably put a beam stop right into the incident beam so that the detector does not get completely oversaturated where the incident beam hits the detector plane. Then exactly for this angle here where Q equals the reciprocal lattice vector that is drawn here, then at that point on the detector plane, we will expect to see a maximum in the scattered light intensity. Yeah, this connects this concept of the Ewald sphere in reciprocal space, which is maybe a bit abstract if you're not so familiar with the reciprocal space, but it connects it right with basically the experiment where the X-ray beam shines on the crystal, and we detect the intensity pattern on a detector plane. And yeah, this is exactly then what you measure when a incident beam hits the crystal target. And now the question is, I mean, here, shown here, okay, I mean, here you would see many, or what we see is many bright spots on the screen. Whereas in this construction here, the Bragg or the Laue condition is actually only fulfilled for this one specific angle, right? Where the k out just points onto one lattice site, but for most other angles, there would be no, this condition would not be fulfilled. Okay, maybe this one here, if it points over here, then here we would probably also get another bright spot on the. So if we want to get this diffraction pattern that shows many constructive interference beams. Yeah, this is recorded using the so-called Laue method, and the Laue method actually, in this case, it's not a monochromatic beam, but here it's a polychromatic X-ray beam, such that not only the angle is probed, but also many different lengths of the incident K vector are probed at the same time. So basically, the radius of the Ewald sphere is varied in a certain range according to the spectrum of the incident beams, and that, of course, increases the number of bright spots that fulfill the constructive interference condition, or the Laue condition. Yeah, so here we measure all diffraction peaks for which this Laue condition is fulfilled for many different K absolute values. Another method, which is called the rotation method, this actually uses monochromatic rays. So here, really, the length of K in is fixed. There's only one K in. But in that case, yeah, actually here it's indicated an angle phi. So in this case, the whole sample is rotated, which would mean that the reciprocal lattice, the whole lattice, is now rotated about this one point here. And in that way, we would also probe the different, essentially, the different lattice planes, or differences between lattice planes that produce then bright spots for specific angles. And this was actually the original method that Max von Laue and his colleagues, Walter Friedrich and Paul Knipping, used in the very first X-ray diffraction imaging experiment, which was performed more than 100 years ago in Munich. And of course, they didn't have a synchrotron, and they, of course, didn't have any XFEL, but it was the good old X-ray tube, yeah, a glass tube. So this actually looks is quite similar to X-ray tubes that are still used today. And they actually made this radiation quite monochromatic by filtering out one characteristic line out of the X-ray spectrum that was emitted from the tube and sent on a copper sulfate sample, and detected the scattered radiation on a photoactive plate. And that was then the diffraction image that they measured at the time. So yeah, of course, not super nice sharp peaks, but they could barely distinguish individual maxima of the intensity pattern here. But so nowadays, of course, it would more look like this. But still, yeah, here in German it says the first X-ray transmission spectrum, or the first transmission signal through a crystal was recorded already so many years ago. Just to maybe also recall from your solid state physics and X-ray scattering course, that there is yet another technique that is also still being used today, which is called powder diffraction. In this case, the sample is not a single crystal, but it's a sample consisting of many micro crystals in powder form, such that the individual small crystals come in the sample with any possible, like with any orientation. And so that basically, so you can view it like this. So we have an assembly of many small crystals that all have different orientations. But what happens is that for one specific incident K vector that has that has a specific length, because we assume monochromatic radiation, and one specific direction given by the beam, then only those microcrystals that just happen to fulfill the Laue condition would produce a scattered bright peak, right? But this happens for any angle in this, like for any azimuthal angle here. So in the end, we get bright counts, not bright peaks. Yeah, and so we get scattering patterns that look more like this. So these images typically are analyzed then by integrating over angle, and then we get a one-dimensional intensity spectrum, just like shown here in red, which then can be analyzed to, for example, learn about the composition of how does the basis of the crystal look like? What is it? A single basis, meaning a monoatomic crystal, or does the basis consist of two or more atoms, like rock salt? Yeah, sodium chloride would be a crystal that has two ions in the basis, or if this is a organic crystal that consists of some organic compound, then like a protein crystal, for example. In this case, every lattice side of the crystal would contain thousands or even 10,000 atoms, and this then produces much more complicated diffraction pattern, obviously. Just to show you that powder diffraction is still being done at synchrotrons and also at free electron lasers, only that the detectors look much more complicated than just a photoplate. Here we have these microstrip detectors. There's many different technologies to record the diffracted X-ray beams around the sample, or these kind of detectors for powder diffraction. This is at the Swiss Light Source in Switzerland. Yes, so that is a brief introduction into the, let's say, classical X-ray diffraction techniques, where mostly it's about recording diffraction patterns from crystalline samples. If we go one step back and calculate, derive from theory, we don't go through all the details, but derive a more general expression for the intensity distribution as a function of the Q vector. The Q, this is always the figure of merit here. That's the difference between incident and outgoing wave vector. Then by going through the math, we get this formula. So this is the important result, which essentially tells us that where the rho here is the scattering density, it's called. So that's the density distribution in real space of the objects that scatter the light, and in any sample, what scatters the X-rays is actually the electrons. So X-ray scattering happens of the electrons in the electron cloud of the atom or the molecules. And what this expression tells us is that the intensity distribution essentially is given by the Fourier transform. Yeah, this is the function times e to the minus IQR d3r. This is the three-dimensional Fourier transform of the scattering distribution, and module square of this. So that's maybe something to remember. Yeah, what we detect as a intensity distribution is the module square of the Fourier transform of the real space distribution of electrons in the sample. Now, yeah, if we apply this theory to just what we have looked at before, just a crystal lattice where the rho here is distributed in terms in the shape of localized scattering centers that are arranged in this periodic way because it's a crystal, then taking the Fourier transform of a periodic assembly or periodic like sum of delta functions, what would you expect to see as the Fourier transform of a periodic arrangement of scattering sites? What's the Fourier transform? So this actually like this is a sum of Dirac delta functions in one dimension would be called a Dirac comb. It's a comb, so it's a periodic arrangement of Dirac delta functions. Maybe somebody knows what the Fourier transform of such a function would be, or any wild guess. What's the Fourier transform of a periodic structure? What's the Fourier transform of a Dirac delta comb? Any ideas? It's very much equivalent to, let's say, so those of you who are familiar with femtosecond lasers, there you know that the time domain and frequency domains are related by Fourier transformation, and femtosecond laser, like femtosecond laser oscillators, produce these trains of femtosecond laser pulses in time. So what do you get as a Fourier transform of a pulse train? The femtosecond people should know this. Like what's this, the frequency spectrum of a femtosecond, let's say 100 megahertz femtosecond pulse train? What's the frequency spectrum, which is just the Fourier transform of the pulse train in time? Anybody knows?
It's the frequency comb, right? This like period, just a lot of periodic spikes.
Exactly. Yeah, periodic spikes in time transform into periodic spikes in frequency. Yeah, the pulse train, femtosecond pulse train, transforms into a frequency comb by Fourier transformation. So now you should be able to guess what this crystal lattice transforms into by Fourier transformation to get the intensity distribution. Asbjørn,
Is it also just a lot of periodic Dirac functions then, or?
Yes, okay. That's this, right? This is what we just talked about.
Yes. Okay. Yeah.
And it's just to motivate or to explain this Fourier transform formula here. Of course, still the same discussion applies with Bragg diffraction and Laue condition, only now formulated in terms of this this formula. But yeah, but I mean the main goal of X-ray diffraction is, of course, it's nice to measure all these sharp peaks on the screen, but what you actually want to do is to then back transform from the intensity distribution into the real space distribution of the scattering density, because the scattering density is nothing else than the electron distribution, and that's that gives you the crystal structure, right? In the end, we want to learn about the structure of the crystal. So what we have to do is, from the intensity distribution, in principle, calculate back, so do a Fourier back transformation to get the real space distribution. And now here comes a big problem, because what we measure is not actually the Fourier transform, but it's the module square of the Fourier transform. And I mean, yeah, if you look at the formula for the Fourier transform, there's an e to the IQR. So this is a complex function, and therefore the Fourier transform is complex, and so it's a complex. It's a distribution of complex numbers. Complex numbers have an amplitude and a phase. But if once we take the absolute value square, we lose the phase information. So we cannot, unfortunately, directly back transform by just carrying out another Fourier transformation, because we are missing the phase information. Yeah, because by taking the module square, we lose the phase. And this is a very general problem in X-ray diffraction imaging, that we have to come up with some clever algorithms or some clever protocols how to retrieve the phase, or how to guess the phase to calculate back to the real space density distribution. And this is typically done by an iterative approach, in the sense that we measure some diffraction pattern, and then essentially forward simulate what the diffraction pattern, what kind of diffraction pattern we would expect for a specific electron density distribution that we somehow guess by doing some intelligent guess, which is based on some atomic model. And yeah, from that we would then calculate what the diffraction pattern looks like, compare with the measured one, see that it doesn't perfectly match. So then we would try by some kind of nonlinear fitting or like optimization algorithm, try to readjust the density distribution to make the agreement between the calculated diffraction pattern and the actually measured one, to make the agreement better and better by going around the loop. So this is an iterative procedure. And then, of course, there's many tricks, like what is the most intelligent way of guessing the initial density distribution, and possibly we can actually still identify specific patterns in this diffraction image, so that we know at least what is the composition, like how many nitrogen, how many oxygen atoms, and usually there is these groups of atoms which always produce the same diffraction pattern. So then we can at least arrange certain groups and so on. Yeah, there's many tricks to do it. Just to mention that actually this kind of diffraction imaging for these more complex systems, namely mostly for proteins, is really the most important field of application of synchrotron X-ray radiation. So this is most of the beamlines at synchrotrons do just that. And what is shown here is like this reconstructed density distribution for one important protein, this GROWEL protein here, that was measured by diffraction imaging at different levels of resolution, from 4 angstrom up to 32 angstrom. Of course, 32 angstrom, this is quite bad resolution. So there definitely we do not capture the atomic resolution of the protein. 4 angstrom is just about getting there to the atomic level. And just to show you the resolution. Now again, I don't see. This is, I think, photon energy. So if we really want to get down to an angstrom resolution to get onto the atomic level, which is like here around 1 angstrom, then we need 20 keV of photon energy, and we need to be able to detect the scattered radiation up to quite large scattering angles. Yeah, that's another important thing. If we like the further out in angle we detect, the higher the resolution and the more information about the small structures we get. Whereas what is called small angle scattering, which would be the scattered intensity around 0 scattering angle, this does not inform about atomic resolution for sure, but for very small scattering angles, then we get more information about the like large scale variations of distribution, for example, the shape of the whole protein. Yeah, then for these complex structures like proteins, which have like some parts of the protein does have a regular structure, but the overall distribution is certainly not like a monocrystal. Then this produces very complex scattering patterns because it's a complex structure. And then there's many tricks for doing this phase retrieval. One, as I mentioned, is trying to apply some intelligent guessing of the structure, and like based on some pre-knowledge about the composition, about structural motifs, and so on. What always helps is to tune the photon energy, in particular to tune the photon energy over specific ionization edges, like the carbon K edge, nitrogen S edge, phosphor edges, if there's any phosphor in the protein, because close to the ionization edges, then the scattering amplitude of these specific elements would be enhanced. And then, yeah, we get different information about the arrangement of the specific atoms in the protein. So we can more like select out specific elements that and to get their structural information in the protein. So that helps. We can produce different samples by replacing certain atoms with high Z. High Z means heavy atoms that contain many electrons, because it's the electrons that scatter. So an atom that contains many electrons, like a heavy metal, for example, or iodine, or like some heavy marker atom, would then scatter the light much more efficiently. What also always helps is to record many diffraction patterns of the same sample, so oversample, and produce redundant data. And then there's numerical procedures that can be applied to get the most information out of these scattering images. And yeah, in this way, since really this is the most important application of X-ray beams overall, it's interesting to see what has been achieved. And so this shows here a number of deposited macromolecular structures, and mostly protein structures, in the protein data bank. So there's this international database that contains basically all the information about the known proteins. And the yellow here, this shows the number of deposited structures of proteins recorded by X-ray diffraction over the years. And okay, it goes up to 2017 or so, but for sure this number just has been increasing since then until today. So X-ray diffraction of macromolecules is very important. And just to give you also one more timely example of X-ray diffraction experiments, this was one that was carried out at DESY in Hamburg at the synchrotron there. And that was actually carried out 2021. So this was just the COVID time. when not only people, when we were all affected by COVID, and even also X-ray science was affected by COVID, and there were actually specific dedicated user beam times only for COVID research. So those scientists who had good ideas to do research on COVID virus that was the best time to apply for beam time, because you had very high chances to get a lot of beam time to study the COVID virus. And yeah, here this shows some, without going into the detail, this was just a very systematic screening study of there are basically many different known biomedical compounds that were screened systematically to see to find some approach how to tackle the SARS-CoV-2 virus. And I think it says, in a search for a drug against COVID, we have performed high throughput X-ray crystallographic screen of 2 repurposing drug libraries. So the full libraries of drugs have been screened. We identified 2 allosteric binding sites represent attractive targets for drug development. So this is an example where there's more basic research facilities, like the Petra synchrotron in Hamburg was actually used, and I'm not sure if in the end the study helped to develop a real drug, but at least this gave some information about what kind of drugs could be efficient for to fight against COVID. All right, but now let's get back to XFEL. And the main difference is that, as I said, XFELs produce X-rays, but with main difference, much higher intensity and femtosecond pulses. And that causes one fundamental issue, which is illustrated here, namely that if such an intense free electron laser X-ray pulse now hits a target, in particular some biological sample, then this is what happens to the sample. Then since it is highly ionizing radiation and it's so intense, then within the pulse that hits the target will be ionized and decompose. And if the pulse is quite long, like the white dots, which represent photons in this pulse of X-ray photons, then you will see that actually during the X-ray pulse already the target starts to decompose. And obviously this is not ideal if we want to record a bright and high contrast scattering image from these photons that are supposed to scatter from the target. If the target already starts decomposing and the atoms become ions and start moving away during the pulse, then obviously the contrast and the structure of the scattered X-ray pattern will be smeared out and the contrast will be very low. So what is much better is a pulse like this. So short that it completely interacts with the intact sample before much later than the decomposition of the target sets in. And this is taken from a website from LCLS, from the LTLS XFEL in California, because it was the first one to generate these femtosecond intense X-ray pulses. And so this is one way of addressing this issue of diffraction before destruction. You have to make sure that the light interacts with the intact sample before it gets destroyed by ionization and Coulomb explosion. But before X-ray diffraction before destruction at LCLS, actually it all started with the first soft X-ray free electron laser, which was the first SARS laser of all. Yeah, this was the flash laser in Hamburg. Not hard X-ray, but only soft X-ray. But so the resolution for soft X-ray obviously is much lower than, because the wavelength is longer than for LTLS hard X-ray pulses. So what they started doing to demonstrate the diffraction imaging, like femtosecond diffractive imaging technique, was to use a nano target, but not like a free, let's say, protein and also, but this was a like nano-fabricated thin metal target supported in a frame, and then by shooting one femtosecond soft X-ray pulse onto this target and then recording the diffraction image on a CCD camera, they recorded patterns like this. Of course, and then from this, they were able to reconstruct by this phase retrieval algorithm. They were able to reconstruct the real space distribution like this, which matches quite well. The actual, I think this is probably a electron micrograph here from an electron microscope. Of course, this method works only on a single shot basis, because a single femtosecond X-ray, even if it's soft X-ray pulse that is intense enough to create a whole scattering image, is intense enough to destroy this sample. So actually, they had a whole array of these samples, and then for every laser shot, they had to move in a fresh sample and then record another scattering image. So that's, of course, the drawback of femtosecond X-ray diffraction imaging. The pulses are so intense that every shot destroys the sample. And this makes it much more complicated to do experiments on these supported targets, because then you have to really mechanically move in and out the next target. Therefore, it's very advantageous to have targets that are renewable, like, for example, these silver nanocrystals that are produced in a magnetron sputter aggregation source to produce metal clusters, because then this source just continuously spits out these nanocrystals in a jet in vacuum, and we have plenty of fresh targets for every free electron laser pulse here. There, only the disadvantage is that of course, these particles come at random times, and they also have they come in a jet that has a certain width. So many free electron laser pulses probably miss the crystal, and only every once in a while a pulse happens to just hit the target and then flash. Then we get this bright image on the screen. And you can see that here. This just again illustrates the Laue condition for diffraction. So here on the left, these two images here, these are actually experimentally measured diffraction images for these nanocrystals that just happen to come at the right time, and that have these different crystalline structures actually. So this is a icosahedron. This is another structure here that produce these diffraction images. On the right-hand side, these are then simulations, basically, by guessing what the structure would look like, then simulating how the diffraction image looks like, comparing with the measured diffraction image, and repeating this by optimizing the structure, trying out different structures until they find the best match. You can see that this is quite a good match, but not perfect. So, meaning that the actual nanocrystal probably looks slightly different, maybe has some defects or so, but overall the match is quite good. and this icosahedron then would produce such a diffraction image like this. So you can see this is actually a combination of periodic things, because I mean it is a crystal, and a nanocrystalline, so there is some periodicity in the structure, but of course it's not an infinitely extended perfect single crystal. Therefore, we cannot expect this perfect regular pattern of peaks, but it has this specific shape, which comes from the finite size and the specific shape of the particle. So to get a better understanding of that, let's see here. This is from Wikipedia. You can just download it. Let's see how the far field diffraction patterns look like for just very simple two-dimensional geometrical structures. So for just a circular aperture, then we would expect by Fourier transform, we would expect this kind of single maximum. This is, you probably know, this is like diffraction of a very small circular aperture. If we take 2, let me see, can I stop this? Anyway, let's just see how these different patterns transform into the far field diffraction pattern. Let me see, can I stop this? Yes. Yeah, for a triangle, let me go back and stop. Yeah, for an arrangement of 2 apertures, this is already, let's say, the first step towards a regular periodic lattice. Yeah, then the diffraction starts looking more already a little bit periodic. If it's 3, yeah, then it becomes even more periodic. If it's, let's see, if it's 4, yeah, this is more like the crystal lattice. This looks more like this the lower diffraction pattern. If it's a large circular aperture, then we expect to see not just one bright maximum, but we expect to see these diffraction rings. Then if it's hollow, yeah, okay, this is all again rings, but this still, of course, has circular symmetry. For a triangle, we get structures like this. So this looks more similar to the icosahedron, because yeah, you can see the icosahedron has this triangle as a basic geometrical structure. For a square, it's more square-like. Yeah. Okay. Then also these, because these rectangles, then it becomes more complex. Anyway, so this is, you can look it up for yourself. This is from Wikipedia. Just Google or look up in Wikipedia Fraunhofer diffraction. That's the far field diffraction. Just want to point out the diffraction pattern once again of a large circular aperture. And remind you a slide that I have shown, I think, the last time, where this is was an experiment where actually me and Shiva Ramakrishnan was also involved at the European Free Electron Laser, where we recorded coherent diffraction images from helium nanodroplets. And turns out that these helium nanodroplets have many interesting properties. But one property is that these helium droplets are really nearly perfect little spheres with size ranges between, let's say, 10 and 100 nanometers. So this, if we want to just test the diffraction imaging method, then helium nanodroplets are really very well suited target systems because they are these perfect spherical shape. And I showed last time these diffraction images that we recorded at the time, which look exactly as expected or very similar to the two-dimensional circular aperture. Just as a recall, at the time, the experiment was done not just with a monochromatic XFEL pulse, but actually this was a pair of two X-ray pulses with different photon energies, and therefore, actually, if you analyze the radial profile by integrating over angles, we could see that there was this beat pattern that's shown in this little insert here, and this beat pattern, like these two superimposing frequency components here, this comes exactly from the two colors of the two laser pulses that both almost simultaneously interacted with the droplets. And then for the first time, we were able, using this very specific detector, to actually decompose this and decompose it into the red X-ray photon. So this is both X-ray photons, but one less energetic, one more higher energetic. And this it's quite an exciting technique because this will hopefully in the future allow us to do actual pump probe experiments with like two time delayed X-ray pulses and then study the dynamics or the structural dynamics of not just spherical helium droplets, but also more complicated and more interesting nano targets. Yeah, just to mention also Asbjørn or other people that are actually into this business for a perfect sphere, a homogeneous sphere, the diffraction, the radial profile is actually given by an analytic expression. You can directly solve the Fraunhofer diffraction integral for a spherical homogeneous distribution, and then we arrive at this analytic formula, which is shown in red. And that obviously matches quite well what we also measured in the experiment. Yeah, just to give you a few more examples of this now femtosecond single shot X-ray diffraction imaging. Another way of delivering renewable targets. So it's and in particular targets that are maybe a bit more interesting than just helium nanodroplets, because helium nanodroplets, you don't find them too often in nature. Actually, they do not exist in nature. But many people are interested in things like proteins or viruses or like biological systems that are actually very prominent in nature and very important. And oftentimes these biological nano systems like viruses or bacteria or so actually live in an aqueous environment. So they are solvated in water. For example, in your body, proteins and biomolecules are usually surrounded by water. So it might be a good idea to try to actually produce samples, not as free nanoparticles, but solvated in a water jet or in a liquid jet, so that we can study their structure in really a natural environment. And this can be done. You can actually produce a jet. This is called the liquid microjet technique. So you can produce a jet of a liquid that is ejected into the vacuum to then interact with a X-ray pulse or X-ray beam, and then measure the diffracted radiation. And the most versatile approach is to use two detectors, one placed closer to the target so that we can detect the wide-angle scattering. And wide-angle scattering have the large theta values. Remember, this is what informs about the small-scale structural variations, so up to atomic resolution, whereas the small angle scattering, small angles means small q values, means large real space variations. This informs more about the overall shape and size of the nanoparticles. This is a photograph showing the moment of interaction of an X-ray beam with such a liquid microjet. I mean, these X-ray beams are very intense and they are highly ionizing. So obviously an X-ray beam that hits the water jet ionizes it and explodes it, which can be seen really as these like flashes and basically cutting a hole into the jet. But since the pulses are short enough, still we are able to record diffraction before destruction. At least that's the hope, to do diffraction before destruction, so to get the scattering information before all this splashing and destruction of the target happens. Yeah, just to give an example of a scattering image recorded from the first detector that records the wide-angle scattering. There you can then typically like identify specific groups, which can be attributed to like really the atomic crystalline structure inside the nanoparticle, whereas here at the small angle scattering, these patterns here, this can be related to the overall shape. So this is more sizes on the length scale of hundreds of nanometers. So this informs about the shape and size of the nanoparticle where the diffraction happened. But overall, then the goal is to like reconstruct from these different scattering patterns to reconstruct the real space distribution. And for example, here, these diffraction images were recorded for a very important protein pigment called photosystem 1. So this is a pigment protein that is important for photosynthesis in I think in algae and like primitive plants, but so actual importance for in natural systems. And yeah, just to illustrate this diffraction before destruction aspect. So here the pulses were varied in their pulse duration from 10 femtoseconds up to 200 femtoseconds. And what we can see is like for the different Q values, which is like Q is proportional to the scattering angle. So at small scattering angle, so small Q, meaning the large scales of the sample, there is basically no difference between short pulses and long pulses, meaning that it doesn't really matter whether it's a long pulse, but for the high Q values, meaning the small scales, there we can see that the contrast goes down for the longer pulses, which means that already during the 200 femtosecond pulse, the target starts to decompose and to lose the structural information, and so the target gets damaged by radiation damage, and scattering intensity drops on the small atomic scales, which are measured at large Q values. Another very prominent examples. Yeah, so these are just a few examples taken from Nature and Science publications. Here, this is the first diffraction image of a virus, a very large one, though. The Mimi virus, I think, is a particularly large one, so that like more than 200 nanometers. But and yeah, why large? Because the resolution that we have with today's free electron laser pulses is still not really at the level of atomic resolution for single particle imaging. So the larger particles are, of course, easier to resolve. And yeah, at least it was possible to get some structural information about these mimiviruses on this scale of tens to hundreds of nanometers. Or another interesting example is here, in this case, using an aerosol sample injector. They injected a beam of living cells into the vacuum and intercepted these cells with X-ray pulses to detect the scattering images. And yeah, so this shows then a collection of different individual snapshots for different cells that show that, of course, like in general, the larger the system becomes, the more variation every single particle has from one particle to the next. And this is another great complication for this single particle diffraction imaging, that usually you have very strong variation from shot to shot. I mean, you can also see here the individual snapshots produce quite different scattering patterns, and every single scattering pattern needs to be analyzed so that hopefully we can retrieve with enough reliability, we can retrieve how the actual shape looked like. Or another very recent example here was actually single protein molecules. This is again the GROWIL, this protein. So again, with a nanoparticle injector, it was possible to inject into the vacuum individual proteins and then record scattering images from these individual protein molecules. Yeah, so I mean, nowadays one of the latest developments in free electron laser science is that these free electron laser pulses now come at very high repetition rate, up to a megahertz repetition rate, and that is good news for these single shot imaging studies, because I mentioned that every single shot looks different, even for nominally the same type of bacteria in this case. So the only way to like really get systematic information about these systems is to just record many single shots of these samples. I mean, of course, always a new fresh sample, because this bacteria will be completely exploded after interacting with one laser pulse. So yeah, then we need to be able to refresh the target and produce many target systems of the same kind and record many diffraction images. So this is called serial femtosecond X-ray diffraction. Yeah, so there's now more and more also numerical and computational efforts that's needed to cope with these enormous amounts of data when these thousands and hundreds of thousands diffraction images are taken. And so this basically here's a very interesting review article called in this Nature Review Methods Primers journal. It's a quite new journal that basically compares the good old conventional rotation crystallography, where the crystalline sample was put into the beam and then rotated according to this rotation method to record the diffraction patterns for different angles. Nowadays, it would be injecting many target nanoparticles into the beam and recording these many serial diffraction images, and then by doing some complicated mathematical analysis, try to retrieve the structure of all these particles. So that's this serial diffraction, serial femtosecond diffraction imaging. Of course, requires many single particle images, requires then classification methods. For example, for classifying according to the different orientations. Then one can average over one class for one specific orientation and then combine all these information for the different orientation and then try to, in this way, reconstruct the full three-dimensional structure. But this needs many identical samples and many diffraction images. So that's probably the most important new trend now in these free electron laser based diffraction imaging experiments that take advantage of the high repetition rate of the modern XFELs. All right, this is what I wanted to tell you as a primer to coherent diffraction imaging. So tomorrow we will hear two more presentations about recent experiments where the single-shot coherent diffraction imaging was actually used on different target systems. And are there any questions to this topic? Questions or comments? Everything clear or everything unclear? That's always the question. Okay, I hope it is more or less clear.
Yeah, most of it was clear. Yes.
Most of it is clear. Very good. Thanks for the feedback. Then, if there are no questions, then I would say we stop for today. So tomorrow we'll hear more examples of this coherent diffraction imaging with femtosecond X-ray pulses. And then I wish you good night and see you again tomorrow. Bye-bye.
Thank you.